This assignment will highlight the Biotech Academy's free virtual lab that utilizes yeast to make human insulin from recombinant DNA. Use the Chrome browser. Enter this URL. Select the Biotech Academy. To change the virtual lab to English, go to the bottom right-hand corner. Select the United States flag. Select the Production of Insulin button. First time into the lab, it is definitely worth exploring what is in the lab. For today, though, we will select no thanks for the laboratory tour. The phenomena behind the Insulin Sciventure is that student scientists will enter this virtual biotechnology lab where they will be challenged to use their skills with transcription and translation to have yeast create human insulin. To successfully complete the challenge, they will essentially replicate the pioneering work behind Eli Lilly's introduction of Humulin, which was FDA approved October 1982. Go to the bottom left-hand section. Select the clipboard. This is the experiment guide or the instructions. The steps that are listed here will guide the student scientist's actions in the Biotechnology Lab. When each step is completed, the platform itself will provide a check mark. To go back to the laboratory, in the upper left-hand corner, click the back arrow. Also important is the inventory section. This holds the equipment you select to bring to the lab. Within the lab itself, the stock area and the refrigerator hold the supplies that you will need to select to bring to the lab. Once again, we're going to go back to the guide. Step 1 is to design a DNA that will allow yeast to make insulin. Click on the door. From the pop-up window, select the computer. From among the options, select Design DNA. For this step, it would be good to provide students with, or ask them to find for themselves, a resource that lists a gene by its parts. Each of the possible components are on the left. Remind students that they will not need all components for this one piece of DNA. Here is one key piece of information. Without the promoter, the yeast will not make insulin. Add the promoter. Click on it and then drag it in to the building DNA region. Click on Insulin. Drag it to the design space. Follow a similar process to add antibiotic B. There needs to be a stop between the two genes. Add the stop. To move the stop between the genes, use the left arrow. Each of the two genes will need a start and stop signal. The overall DNA strand will need a promoter, then a ribosome binding section. The end of the DNA will need a terminator region. Build this recombinant or designer DNA. I strongly suggest having all teams show you the DNA design before going back to the lab. There is something to be said for troubleshooting from failed experiments. However, if the recombinant DNA is not constructed properly, the insulin protein will not be made. Within the virtual lab, it will take students quite some time to realize the DNA construct had failed. If we look towards the bottom, the sequence information is actually color-coded to match the different segments of the DNA construct. So if we scroll through that, we will see the DNA sequence. So for the blue, for the insulin, for the gene, this would be the insulin sequence. Right before that, in light green, is the start codon. This is a good place for students to be able to review and understand that the chunks up here are sequences of nucleotides. It's very important when the students have completed their designer DNA that they click Order before returning to the lab. Then once again, it is the back arrow to return to the lab. Returning to the lab, we see that the DNA construct is in a test tube in the inventory. Select the guide. Notice that that first step has a check mark. For our next step, we're going to have to mix yeast and our DNA construct together, and then we're going to have to put it into an instrument called an electroporator, which will poke holes inside the cell membrane. So to return to the lab, back arrow, we're going to go to the stockroom. Select the bottom drawer. Add a test tube to the inventory. Use the back arrow. Select the refrigerator. Add yeast cells to the inventory. Return to the lab. Check the lab assignment for directions. We will mix the yeast and the DNA in a test tube and then add the sample to that instrument. Now we're going to go to the lab to complete the work. Return to the lab, select the laboratory table. First step is to light the Bunsen burner. Simply click the top of the Bunsen burner. Next, add our test tube from the inventory and move it to a test tube rack. Move the yeast test tube to the test tube rack. In the bottom right-hand corner is a pipette. Click on that pipette, drag the pipette to add a tip to it. Click and drag the pipette to the yeast sample. Add the yeast sample to the test tube, then back to the bottom, dispose of the used tip. We're going to repeat this process to add a sample of the DNA to that test tube. We are now ready to add a sample of the yeast DNA mixture to the electroporator. Select the new tip, collect the sample, dispense the sample onto the instrument, discard the tip, return the pipette to its holder, select the dial on the electroporator. The movie that loads is illustrative of what is happening within that test tube. Student scientists see the DNA enter the cell. The DNA is then transcribed to messenger RNA. Nucleotides are attached one by one as the messenger RNA grows. Next, the messenger RNA is translated. The ribosome assembles around the messenger RNA. The protein is assembled one amino acid at a time. The protein folds to its three-dimensional shape. At the end of the movie, the platform says one has successfully had the organism make the proteins. Back within the main lab, select the guide. This is where the high school assignment will end. Student scientists have essentially completed everything they need for this to be illustrative of DNA to protein curriculum. The additional steps are needed to complete the isolation of the human insulin protein. However, such level of detail is more applicable either as a project for extension or for AP Biology curriculum level. Steps 3 through 8 are selection of the genetically modified yeast cells and optimization of growth conditions. A batch of yeast that is making human insulin is grown. Step 9 is the isolation of the human insulin protein. For educators thinking ahead to the LS3 curriculum, please note that this biotechnology virtual lab has an additional assignment where student scientists can explore the role of insulin in glucose homeostasis. This completes the Sciventure NGSS and Beyond implementation movie for the Biotech Academy's virtual lab on insulin production.